Forensic Science Exam Prep presents Diatoms. Diatoms are microscopic unicellular water algae with a rigid case composed of silica which belong to class Bacillariophyceae and said to enter the pulmonary circulation during drowning. There are numerous forms of diatoms found in different shape and size both in fresh and sea water. If the life is still present, the heart will be beating and will transport diatoms which penetrate the lungs, living to distant parts of the body, brain, bone marrow, etc. In a corpse this cannot occur, thus presence of diatom in bone marrow is a sure sign of antemortem drowning. Diatom examination. Importance. In drowning deaths, whether death was due to suicide on homicide, observation of diatoms is significant proof of drowning. In the postmortems carried out on bodies of drowning victims, detection of diatoms in the autopsy signifies the cause of death. If the drowning had taken place in the fresh water, i.e. ponds, lakes or rivers, then freshwater diatoms are found in the autopsied body. Presence of diatoms in the body is not a conclusive proof of drowning death. However, in many instances, when postmortem was conducted in accident deaths, diatoms were found in the autopsied bodies. Hence, presence of diatoms is not a conclusive proof of drowning. The nature of diatom found in the autopsied body indicates the site of occurrence of death, i.e. if freshwater diatoms are found in the body, drowning had taken place in fresh waters, i.e. ponds, lakes, rivers. If marine diatoms are recovered in postmortem, then body was drowned in ocean. Method. Before diatoms can be examined, they have to be cleaned. This involves the removal of cell contents, pigments, sand, mud or other material likely to interfere with microscope examination. A sample of material may conveniently be obtained from femoral or sternal bone marrow. Care should be taken to use dry and clean instruments to avoid any contamination from extraneous material. After removing the sternum, a small window is cut into it, and marrow exposed. A small portion of the marrow is scooped out into a test tube. Acid digestion of the marrow is then effected by mixing it with strong nitric acid, and heating it till a clear fluid is obtained. This is cooled, then centrifuged, and the sediment is examined under the microscope. A remarkable collection of most delicate and beautiful skeletons of diatoms is seen in cases of death from drowning in favorable circumstances. Even the site of drowning can be determined, since the freshwater and the seawater diatoms are different, and the seawater plants even vary from place to place along the coast. The diatom test is valid if it can be shown that the deceased did not drink this water immediately before submersion. The species recovered from the specimen are all present in the sample from the site of drowning, and the various species are present, if sufficient are found, in the same order of dominance. For the admissible size range and in approximately the same proportions. Since diatoms resist putrefaction, the diatom test is particularly valuable where decomposition is advanced. Diatom test is negative in dead bodies thrown in water and in dry drowning. Standards and controls. In diatom examination, invariably the control water samples must be used for comparison purpose. Standard diatom samples can be preserved on slides and can be used as standards for comparison purpose. Diatom report format. Diatoms were detected, not detected in the Exhibit S, and were comparable, not comparable, with the control sample Exhibit S. Medico-legal issues. The presence of diatom indicates antemortem drowning. While doing post-mortem, if diatom is found in intestines or in the stomach, it clearly indicates that the person was alive when he entered into the medium. It can also be found that if the person was first killed and then pushed into water. For instance, the diatoms will not enter the intestines or stomach if he was killed and then pushed into water. The diatoms would also show if the body was dragged out of water, etc. The diatom test is valid if it can be shown that the deceased did not drink this water immediately before submersion. Diatom test is negative in dead bodies thrown in water and in dry drowning.